So, okay, students, good afternoon, all of you. Please on your videos. Can you hear me? Please on your video. Please, all of you on your video. On your video. Okay, Lakshmi, please on your video. Okay. On your video. All of you on your videos. Right.
Good afternoon, Miss.
Good afternoon. Shifana on your video. Shamina, please on your video.
Good afternoon, Miss.
Good afternoon. Okay. Okay, so today we can start our class. So we are beginning with microbial genetics. I hope all of you can see the screen. Okay. Can you hear me?
Okay, Miss.
Okay.
Yes,
So,
Miss.
okay.
Okay.
Okay, right. We'll begin with microbial genetics. This is the first part of microbial genetics. So in microbial genetics, we have to study the life cycle of retrovirus. So in today's class, we are dealing with the structure of retrovirus, not retrovirus, in general virus. Okay.
Okay, Miss.
Okay. This virus, it is classified in different ways. The genome of the virus, the classification depends on the genome of the virus. Then whether it is the genome is single stranded or double stranded, the sense whether it is positive sense or negative sense, method of replication, everything determines its class. So the virus, it is classified in different ways, depends on the genomes, viral genomes, nucleic acid, whether it is DNA or RNA, then stranded, that is single stranded or double stranded, positive sense or negative sense, method of replication, everything determines the classification of virus. And other classification is determined by the disease caused by the virus or virus morphology. So according to one classification, this virus can be placed in one of these 7 groups. That is, in group 1, there are double-stranded DNA virus. This is one classification. In this classification, the group 1 includes double-stranded DNA virus like adenovirus, herpes virus, pox virus, etc. And in group 2, it includes positive sense single-stranded DNA virus like parvoviruses. And group 3 includes double-stranded RNA virus. They are the reoviruses. Group 4 includes single-stranded positive sense RNA virus. Group 4 includes single-stranded positive sense RNA virus. And group 5 includes single-stranded negative sense RNA virus. And group 6 include single-stranded positive sense RNA virus which causes reverse transcriptase enzyme. Here comes the retrovirus. That is, group 6 includes the retrovirus. And group 7, in group 7 there is double-stranded DNA virus with reverse transcriptase enzyme. So this is according to one classification. There are 7 groups, and in these groups, in group 1, it includes double-stranded DNA virus. Group 2 includes single-stranded DNA virus, but it is positive sense single-stranded DNA virus. Group 3 includes double-stranded RNA virus. Group 4 includes positive sense single-stranded RNA virus. Group 5 includes negative sense single-stranded RNA virus. And group 6 includes single-stranded RNA virus with a reverse transcriptase enzyme. And group 7 includes double-stranded DNA virus with a reverse transcriptase enzyme. So this is one classification, and we are focusing on retrovirus. So what are retrovirus? Retrovirus are diverse family of animal virus that contain RNA as their genetic material. So this retrovirus, they contain RNA as their genetic material. It's a diverse family of animal virus. Retrovirus are diverse family of animal virus, and it contains RNA as their genetic material. But they produce a double-stranded DNA copy of their genome upon infection. This retrovirus, they contain RNA as the genetic material, and they produce a double-stranded DNA copy of their genome upon infection. So upon infection in the host cell, they produce a double-stranded DNA copy of their genome. So such viruses are called retrovirus. So retrovirus is a diverse family of RNA virus. They contain RNA as the genetic material, and upon infection, they produce double-stranded DNA copy of their genome in the host cell. And this dual genetic system allows retrovirus. What are the advantages? It allows the transmission of virus from one cell to cell as packaged RNA. So it helps the virus, the transmission of virus from one cell to another, or one cell to other cell as packaged RNA. And when this retrovirus leaves the host cell, it leaves a DNA copy in the chromosome of each infected cell, where it can be transmitted from one cell to from one cell generation to next cell generation. So this retrovirus, which contain RNA as their genetic material, and these retrovirus, upon infection, they produce a double-stranded DNA copy of their genome. And this dual genetic system allows retrovirus the transmission of virus from one cell to cell as packaged RNA, and also this retrovirus, it leaves a DNA copy in the chromosome of each infected cell. It leaves a DNA copy of in the chromosome of each infected cell, where it can be transmitted from one cell generation to next. This DNA copy can be transmitted, which is left behind in the host cell, can be transmitted from one cell generation to the next. So retrovirus, that is the family Retroviridae, are enveloped. These retrovirus are enveloped isosahedral virus that possess an RNA of about 7 to 10 kilobases. So retrovirus are enveloped isosahedral virus. What do you mean by isosahedral? Isosahedral means 20 faces of equilateral triangle. That isosahedral picture that I have given in the first slide, that is isosahedral. The picture is given in the first slide, that is 20 faces. It has 20 faces of equilateral triangle, that is isosahedral virus. So these retrovirus are enveloped isosahedral virus, and they possess RNA of about 7 to 10 kilobases. Then retroviruses are divided into two classes, simple retrovirus and complex retrovirus. The complex retrovirus is otherwise known as lentivirus or HIV. So retrovirus are divided into two classes, simple retrovirus and also the complex retrovirus. The second class is complex retrovirus. The simple retrovirus, they encode mainly 3 polyproteins. Simple retrovirus, they encode 3 polyprotein: GAG, pol, and ENV. They encode 3 polyprotein, that is GAG, pol, and ENV. Whereas the complex retrovirus encode 6 accessory protein in addition to these 3 polyproteins. So simple retrovirus, they encode 3 polyprotein, that is GAG, pol, and ENV. Where the complex retrovirus, they encode 6 accessory proteins in addition to these 3 polyproteins. So Dia, Ansila Kabir, please on your video. Nasiha, Lakshmi, Girija. All of you, please on your video. Nasiha, Gayatri, Anjmi. All of you, please on your videos. Please on your videos.
On ആയ മിസ്സേ
ആരാ ചോദിക്കുന്നേ?
കാണാൻ പറ്റുന്നില്ല മിസ്സേ.
Adani, I can see you now. Nehina, I cannot. This video is off. Gayatri, I can see you. Binisha, okay. Fatima, where are you? Nehina, Fatima, please on your video. Nehina, on your video. Fatima, where are you there? Fatima, എവിടെ ഉണ്ടോ? ഉണ്ടെങ്കിൽ വീഡിയോ ഒന്ന് ഓൺ ആക്കിയേ. പ്ലീസ് ഓൺ യുവർ വീഡിയോ. ഗായത്രി, ബിൻഷ, നെഹിന, ഫാത്തിമ.
മിസ് വീഡിയോ ഓൺ ആക്കി.
വീഡിയോ ഓൺ ആക്കിയോ? ആരാണ്? നെഹിനയാണോ? ആരാ വീഡിയോ ഓൺ ആക്കി എന്ന് പറഞ്ഞേ?
നെഹിന.
നെഹിന, ഓക്കെ. ആൻഡ് വാട്ട് അബൌട്ട് ഫാത്തിമ? നെഹിന, ലെറ്റ് മീ സീ. Nehina, but I cannot see you. Nehina, എനിക്ക് കാണാൻ പറ്റുന്നില്ല. And Dia also. നെഹിനയുടെ വീഡിയോ ഇപ്പോഴും ഇപ്പൊ കണ്ടു. ഓക്കെ ദിയ, ഇപ്പൊ എല്ലാരെയും കണ്ടു കേട്ടോ. എന്താ ലക്ഷ്മി ചിരിക്കുന്നേ? ഓക്കെ, നെഹിന വീഡിയോ ഓൺ ആക്കി വെക്കൂ. കളഞ്ഞിട്ട് പോകരുത് ക്ലാസ്. So retrovirus, they are divided into 2 classes, that is simple retrovirus and complex retrovirus. Simple retrovirus, it encodes 3 polyprotein, that is GAG, pol, and EMV, whereas complex retrovirus encodes 6 accessory proteins in addition to 3 polyproteins. So retrovirus cause serious human diseases like acute immune deficiency syndrome and adult T-cell leukemia. That all everybody knows. Now we are coming to virion components. Virion components, it include envelope, RNA, and protein. So virion components, it includes envelope, RNA, and protein. Dia, on your video, envelope it is composed of lipids, and this lipid it is obtained from the host plasma membrane during the budding process. As well as it also possesses glycoproteins, which is encoded by the ENV genes. So envelope composed of lipids and glycoproteins. This lipid, they obtain this lipid from the host plasma membrane from where they budded off. And glycoproteins are encoded by ENV genes. Is that clear? And the retroviral envelope serves 3 distinct functions. It provides protection from the extracellular environment via the lipid bilayer, then enabling the retrovirus to enter and exit the host cell through endosomal pathway, then ability to directly enter cell by fusing with their membranes. So these are the 3 functions which are provided by the envelope of the retrovirus. It provides protection from the extracellular environment via the lipid bilayer. Then it enables the retrovirus to enter and exit the host cell through the endosomal membrane, and the ability to directly enter cell by fusing with their membranes. Next is these proteins. It consists of GAG proteins, protease, pol proteins, and EMB proteins. These are the main proteins. GAG proteins, protease, pol proteins, and EMB protein. GAG protein, so group specific antigen. GAG or group specific antigen proteins are the major components of the viral capsid. These GAG proteins are the major components of the viral capsid. And retroviral GAG proteins are responsible for coordinating many aspects of virion assembly. These GAG proteins are very important during the assembly of the virus. So the GAG proteins or group-specific antigen proteins are the major components of the viral protein. They play a major role during the viral assembly. Next is protease. It is expressed differently in different viruses. Then pol proteins are responsible for the synthesis of viral DNA and integration into host DNA after infection. So pol proteins are important or responsible for the synthesis of viral DNA. Pol proteins are responsible for the synthesis of viral DNA and integration of this DNA into the host DNA after infection. Okay. ENV proteins play a role in association and entry of virions into the host cell. So ENV protein plays a major role in association and entry of virion into the host cell. Several protein species are associated with the RNA in the retrovirus virion. Nucleocapsid protein is the most abundant protein which codes the RNA. So this nucleocapsid protein, it codes the RNA genome of the retrovirus, while other protein present in much smaller amounts have enzymatic activity. So nucleocapsid protein, its function is it codes the genome, RNA genome of the retrovirus. So these are the major proteins that we can see in retrovirus, that is GAG protein, protease, pol proteins, and ENV proteins. Next is viral genome. In retroviral, the viral genome is RNA. It exists in dimer as dimer RNA. What do you mean by dimer RNA? Dimer RNA means it is not double-stranded RNA. It occurs as two. It occurs in two copies. It occurs as two. There will be two RNA molecules in one retrovirus. Two RNA molecules in one retrovirus. So it exists as dimer RNA. This RNA in retrovirus, it exists as dimer RNA. It has a cap at the 5' end and poly A tail at the 3' end. This RNA genome, it has a cap at the 5' end and poly A tail at the 3' end. And genomic RNA is produced as a result of host RNA polymerase II activity and by adding a 5' methyl cap and 3' poly A tail. So this viral genome is RNA. It exists as dimer RNA. At 5' end, it has a cap, and 3' end, it has a poly A tail. And genomic RNA is produced as a result of host RNA polymerase II activity, and this genomic RNA, it has a cap at the 5' end and has a poly A tail at the 3' end. And the 5' end includes 4 regions, which are they? They are R, U5, PBS, and L. The 5' end, it has a cap at the 5' end, and this 5' end, it includes 4 regions, that is R, U5, PBS, and L. What is R? R region is a short repeated sequence at each end of the genome. At each end of the genome, you can see this short repeated sequence, R region. At each end of the genome, there is this short repeated sequence or R region. used during the reverse transcription, and this region is used during the reverse transcription to ensure correct end-to-end transfer in the growing chain. So this R region, it is seen at each end of the genome. It is a short repeated sequence, and it is used during the reverse transcription. This R region is used during the reverse transcription to ensure correct end-to-end transfer in the growing chain, to ensure correct end-to-end transfer in the growing chain. And U5 is a short unique sequence between R and PBS. U5 is a short, it's unique sequence between R and PBS. Now what is PBS? PBS is the primer binding site. PBS or primer binding site consisting of 18 base pair. PBS is the primer binding site and it consists of 18 base pairs, 18 bases. L region is an untranslated leader region that gives the signal for packing of the genome RNA. So these are the region that you find at the 5' end. 5' end, it has a cap, and these are the region that you find at the 5' end of the RNA genome of retrovirus. One is R region. It is a short repeated sequence at each end of the genome, and it is used during reverse transcription to ensure correct end-to-end transfer in the growing chain. U5 is a short unique sequence between R and PBS. PBS is the primer binding site and it consists of 18 bases. L region is an untranslated, it is untranslated leader region. It is untranslated. L region is untranslated leader region, and it gives signal for packing of the genome RNA. This L region, it gives the signal for packing of the genome RNA. Now the 3' end includes 3 regions, which are the PPT or polypurine tract, U3 and R. The 3' end, it includes 3 regions, PPT, U3 and R. PPT is a primer for plus strand DNA synthesis during reverse transcription. PPT or polypurine tract is a primer. It is a primer. PPT is a primer for plus strand DNA synthesis during reverse transcription. And U3 is a unique sequence between PPT and R, which serves as a signal that the provirus can use in transcription. So it's a unique sequence. U3 is a unique sequence between PPT and R, and it serves as a signal that the provirus can use in transcription. The provirus can use in transcription. And R, the terminal repeat sequence at the 3' end. It is the terminal repeat sequence at the 3' end. So these are the 3 regions at the 3' end: PPT, U3 and R. PPT is a primer. It is a primer for plus strand DNA synthesis during reverse transcription. So it is a primer for plus strand DNA synthesis. It synthesizes this RNA is converted to DNA with the help of reverse transcriptase. So PPT is a primer for plus strand DNA synthesis during reverse transcription. U3 is a sequence between PPT and R, which serves as a signal that the provirus can use in transcription. And R, the terminal repeat sequence at the 3' end. So to summarize, we can say that in retrovirus, the genome RNA, it occurs as dimer, and it infects host cell with their genome, and then are reverse transcribed into double-stranded DNA. And this DNA is then integrated into host cell genome, isn't it? So in reverse transcriptase, genome is RNA. It occurs in dimer form, and it infects the host cell with this genome. And then this genome, RNA genome, is reverse transcribed into double-stranded DNA. And this DNA is then integrated